Alrighty, I think we're going to go ahead and get started. It is noon. Thank you guys so much for coming today to the October Lunchtime Lecture. This is part one of our two-part series for the end of the year. Today we're going to be hearing from Craig Meyer, Benjamin Durrington, and Dr. Alejandra Vasco, and I'm going to let them do their introductions themselves. This is going to be a little bit of a shorter talk than I think we're used to, in that each speaker is going to have 15 minutes. At the end of that, they're going to have 5 minutes for questions. And then we'll move on to the next speaker. If you could hold any questions that you have beyond that 5-minute window to the end, I would really appreciate it. Also, we are streaming this online and recording, and I'm going to be passing around a microphone. If you have a question, make sure you are saying it in the microphone so I have it officially. All righty. Thank you all so much.
All right. Hello, everybody. Hello. Hello. So my name is Craig, and I'm a herbarium tech. Today, I'm going to be talking about our efforts at AI-assisted transcription. So I've been very fortunate that I've been able to work with Rachel, who handled a lot of our image processing, and Jason, who handled a lot of the OCR and just getting us started with using GPT. And okay, there we go. Brad. So this is intended to be like a case study on using a specific data set inside the All Asia grant. And so I'm going to be talking about a few things. Like, we're going to start off with, why do you care? Why is this interesting? Then we'll talk a little bit about the context of the grants and the scope of the problem. Then we'll talk a little bit about how we built tools to address that problem. And then finally, the results and where we're going to go next. So why do you care? The most immediate reason is that better tools make transcription faster, easier, and more consistent than traditional manual entry. And so for some couch cushion money and a little bit of effort, we got rates that are 3 to 5 times faster than traditional transcription. And the broader reason that's more applicable to everybody here is that this is a really adaptable methodology that can help with a lot of different kinds of problems. Parsing text, data cleanup, data entry, analysis, like any task that requires you to do the same thing over and over again is likely going to be a candidate here. So, and importantly, I can't code, but I do know how to ask an LLM to code for me. So to walk you through the process, I'm going to show you how we got started using my project as an example. So I'm funded under the All Asia Thematic Collection Network. And so despite enormous biodiversity over this huge area, those samples are digitally underrepresented in American herbaria. So by the numbers, to put things into context, the grant aims to create this single portal with 15 million specimens. Now, 3 million of those are in the US, and Brit's part of that is around 50,000. So this is a breakdown of our Asian specimens, grand total about 50,710. The top 5 countries make up 87% of that total, and the Philippines by itself is 59%. Now, fortunately, a lot of our Philippine stuff comes from one single multi-year project, the Philippines Plant Inventory. You'll hear me talk about it like as the PPI. There we go. And that's where we get started. After transcribing for a few weeks manually, I started thinking, hey, there's got to be a better way. There are things that people are really good at. We're really good at eating stuff. We're really good at rationalizing bad decisions. We're really good at pattern recognition. But in terms of transcription, we're kind of hot garbage. My best days of doing transcription were about 100 to 200 in a day. And that's given really simple, really repetitive labels. Once you add any complexity, like Philippines place names or very long verbatim plant descriptions, speed and accuracy suffers a lot. Similarly, people get bored. Maintaining concentration for hours and hours just isn't possible. And then also there's a million edge cases when you're doing transcription, and the labels don't perfectly match up to the Darwin core format that we're using. So even with the same user, even in the same day, you're going to get variability on how those decisions get made. So the nice thing is robots address all of these problems. They have perfect memories. They copy things exactly. They're faster than we are at a lot of this. They don't get bored, and they always follow the rules that you set, so they make consistent decisions. So in building this process, I wanted to make something that human beings do what they're good at, specifically pattern recognition. And it lets robots do what robots are good at, grunt work. And the goal was intended to be achievable. So instead of aiming for something that is fully automated, handles everything perfectly, I wanted something that just leverages human capacity and makes the process a little bit faster. So I want a bicycle today rather than a spaceship sometime in the distant future. And the key is breaking that problem down into several independent steps. And I'll go into a little bit more detail on all of these, but just as a brief overview, first step is we make a copy of that image and then we crop it down to the label. Next, we submit that image to an OCR process and we get unstructured text back, just the text that's on that label. Next, we parse it by submitting that raw text to a large language model, we use GPT in this case, along with instructions on what all of the text on that label means, and we get it back in a structured format. And then finally, we can compare that now formatted data to the original label and then compare it and upload it in bulk. So let's start with the images. All we have at first is a full-sized image that we've pulled and we've stored locally. And if you look there on the left, you can see a good example of a PPI image. Note that the labels are all typed. Each field is labeled, so you can see that there's location, LOC for location. And the fields are consistently placed and separated from each other. So that and the sheer size of this project makes it a really good case for automation. Now, we're also fortunate that the labels and the annotations are almost always here in the lower right corner, so we can crop out all the extraneous stuff. We don't want the text that's up here on the color target, for instance, and making those images makes the quality control a lot easier later as well. So the next step is here in the optical character recognition or OCR step. We used an Amazon product called Textract. It's not free, but it's really cheap. We ran 14,000 images for around $12. There are free options, but accuracy really suffers. And ultimately, once you factor in the cost of somebody going in and fixing those errors, it ends up being a lot more expensive. So on the left, we have our, oh, sorry guys. Okay, so on the left, we've got this original image, and you can see that those red boxes are the outlines of where Textract recognized text as characters. Now, there's a couple things to notice here. We're getting the text on the label that we want, of course, here, but we're also getting the image stamp and the barcode over on the right as well. Up above, you can also see that that Textract also snagged the handwritten text up above the label as well. So we've gotten everything, in as this raw text. And the ultimate product here is a raw text file, one for each one of our images. So we end up with a folder of 1,000 text files. The next step is a Python script called polyparse. Now here we're submitting these raw text files with a prompt to the GPT engine. And if you look close, you can see our prompt. I'm telling the engine what the text is. So you're going to parse unstructured OCR text from a herbarium. As an example, COLL. This is the name or names of the collectors. It will appear after COLL:. It's a lot like setting somebody down and telling them how I want them to sort and classify this raw text. I'm giving the engine clues to context and meaning as well as positioning relative to other text. I'm also telling it how I want that text formatted. It takes about 45 minutes to run 1,000 of these records. And our end product here is what's called a JSON file. That's what this is here. It's a human and machine readable data format that contains the label information about one specimen. And you can see how the fields have been sorted here. You can see collector, number, island, province. Another thing that's down here on the bottom is you can see the tokens that this used to run this one sample. This process is astonishingly cheap. We ran 14,000 records on this for about $13. So it's an incredibly cheap process to run. And now once this is done, once we have these JSON files for all of these, for all of our images, we can put them all into one spreadsheet. It looks like this. Okay, so to recap, we've cropped our images down, we've extracted the text from the labels, we've parsed that text into specific fields, and now we have it in a spreadsheet. Importantly, everything up to this point has been automated. This is a hit the button and go get a cup of coffee situation. It's only at this point where we introduce people back into the process to do quality control. And the method here is to use synthetic labels. We're replicating the look of the original label here with a spreadsheet. And now we're comparing, one label versus the other. And collecting two collector fields, for instance, is a whole lot faster than typing it in manually. It's a lot easier to find errors. We've also trimmed down the cognitive load and the necessity for training here as well. So the training for a volunteer to compare this to that is a whole lot lower than the training to get a volunteer up for quality full transcription. I don't need you to parse out location. I don't need you to calculate decimal degree latitude. I don't need you to decide whether this information should be in locality and habitat. All I really need you to do is make this look like that. And so with this methodology, we can handle a lot of formatting decisions in bulk at the very end. So to recap, we now have everything structured into a spreadsheet. So instead of relying on individual decisions, we can make big bulk decisions about a lot of stuff using spreadsheet wizardry. All the stuff that we talked about earlier, calculating decimal degrees, annotations based on criteria, like standardizing a date format, we handle it all here at the very end. The spreadsheet looks at our raw working sheet that directly reflects the label, and now we can put it into Darwin form. And after that, it is a simple skeletal upload into the portal, and it's done. Okay, so how do we do? On the left, you're looking at a chart of the All Asia Thematic Collection Network records over time. Unprocessed is here on the bottom, and right here is stage 1. That's where we just have the name and a country. Stage 2 is right here. We've completely transcribed everything on the label. And then for stage 3, we have everything on the label and a point on the map. So we have a location. We've simplified that data over here on the right. It's just looking that this is our to-do list. This is how much we started with, and here's where we are as of, I think, about a week ago. Note also that I've annotated out here both of these with two arrows. Polyparse's first successful run here in May, and the last PPI record here in mid-August. One thing I'd like you to notice, look at the shape of the slope of the line. This is manual transcription here. And when we start using unicycle, the slope of the line gets a little steeper until eventually we really start taking off. And by July, we've hit our top speed with 2,800 records in a single week. Okay, so the question on your mind, if you're anything like me, is, okay, that's great, but what have you done for me lately? What happened here? Why did we slow down? And that really brings us to future development. This process, unicycle, was really optimized for one label format. It's rather than a general purpose, one size fits all. And that made a lot of sense because the PPI dataset was by far the single biggest, juiciest target. We could afford to spend a lot of time tailoring that process to one format. And now that those are done, okay, now we've got to work with increasingly smaller and smaller cohorts of labels, each with different formats, each with different information. Now, where we want to go next is to make that process faster and more automated. Another thing we want to explore is cutting out that middle step of a separate OCR process. Now, originally, the GPT engine, when we first started this, could only handle text. That's why we had OCR at first. Just in the past couple of months, or a few months, OpenAI came out with a new version, 4.0, that can handle images as well. Okay, that's really cool because now we can directly submit those image labels, or the images of the labels, directly to the engine for parsing. We don't need that intermediate step. And the neat thing is that when we do that, we're actually submitting it with more information. It can recognize what is part of the label versus what's part of the barcode or what's part of the image stamp. And that makes that process cleaner. And the last thing that I'm really interested in is making that QC process better. The QC process with synthetic labels is still kind of the weak link in the process. We are torturing a spreadsheet into being an application that it really wasn't designed for. Now, we're working with a former intern, Grant. A lot of you have met him. He's a great guy. To develop a general purpose synthetic label tool, something that's a bit more omnivorous, as part of an entrepreneurship program at TCU. It's very early stages still. We're still just talking about it, but it's really exciting. So where I want to land here is on the iterative nature of the project. You can see the same prompt here given to ChatGPT of 2022 and GPT 4.0 from just a few months ago. We started just by trying to make tools that make transcription easier and by doing it by hand. And then we made it a little bit better. And then we made it a little bit better. GPT helped us in a couple different ways. It parsed the data that we submitted, certainly, but it also wrote most of the scripts that we used. So yes, we made a faster pipeline for transcription, but what I really want y'all to take home is the possibility of having an LLM write you code or do grunt work that helps you focus on the best and highest use of your time. Start small with a limited project and then make it a little bit better. So Jason and I are going to be teaching a brief intro tomorrow, but if you have a specific application or problem, I would really love to chat about it with you sometime. So, and with that, I'll happily answer any questions that y'all have. Thanks.
I have a question for you, and I'd be interested in what the policy is for the Brit Herbarium. In the old days, let's say up until about 1940s, collectors wrote their labels out in script in their handwriting.
Yeah.
There was an advantage to this because you could be sure that they looked at the specimen if they wrote the label. Now the labels are typed. And you can scan those labels, and type labels will give you a more accurate picture because sometimes the scanner can't read the handwriting.
Right.
Do you realize the importance of handwritten labels? And what's the policy now for Brit when it comes to labels?
I mean, for this process, I would say that, it's the quality of the transcription is only as good as the quality of that label. Like, I can't really speak to what was going on in, when they were transcribing the label. So the best that we can do is, translate that label and make sure that our data accurately reflects that label. Am I answering your question?
No, not really. Because you still have to deal with the older specimens and handwriting. So what's your approach to that to make sure you have an accurate label and you can reproduce it? Because there actually is a website now where they have those old collectors and their script handwriting so you can match it up. USDA has that.
Okay. So how are we handling handwriting?
Right.
Okay. This process works best on typewritten. That's why we kind of focused on using the PPI first, because since it's typed, that's easier. We have explored a little bit with using like the direct image submission to handle handwritten notes. I think with some field notes that are handwritten, and the accuracy is lower. It's not as good. So it's going to require a lot more human attention. But even if this handles some of the heavy lifting, I'd say that that's a net benefit.
Well, are the notes actually on the herbarium sheet for, let's say, habitat details, or is that a byproduct and you have to interpret it?
I mean, in this case, I'm just working with labels.
Okay.
So, but yeah.
I think we have time for one more question. All right, great talk. Thank you. I just had a quick question about the text that feeds into the labels. So for example, in the PPI case, you have labels that are computer generated and printed. So presumably there is text data in a file somewhere that generated that text label. Is one of the challenges just a fact that maybe the text file that was used to generate that label, was it not properly databased and backed up and stored somewhere on a computer. And so we're having to now go back and do it this way. So that's maybe the first part of the question. And then the second one is a little bit unrelated, but I'm just kind of also curious if you've played around with, for these more troublesome labels, thinking about benchmarking with different scales of problematic labels so that you can maybe think about assigning confidence scores to when you start to iteratively test the algorithm in terms of its ability to recover text from what maybe you might a priori be able to assign as a different difficulty score, like from 0 to 10, where 10 is my handwriting, which is very difficult to read, and 0 is a, printed large font label, something like that.
Sure, that's a really good question. As far as to the first part of your question, I think that boy, wouldn't it be great if we just had all of this in text somewhere? But I think that is just lost to the mists of time. I think so sadly it's gone. We don't have access to it. But to the second one part, there's actually a really, just to say one part about this, these red boxes that you saw on the text. Oh, so I'm doing it on mine, but not the other one. Okay, so these red boxes here, each one of those red boxes, we actually get 2 products back from TextRack. First is a raw text file that just looks like this, but we also get another JSON file that has information about each one of these text boxes, along with the original text, the location of this box, and a confidence interval, confidence in it. So it is possible for us to use that as well as a proxy for, hey, we don't want to have anything that's lower than an 85% confidence, or we want to flag that in some way. So yeah, there are tools to do that. Cool. All right.
That's all the time we have for questions. Thank you, Craig.
Thank you very much.
Next up is Ben Durrington.
Hello, I'm Ben Durrington, Plant Records Coordinator at the Fort Worth Botanic Garden, and I also assist with curation tasks of the living collections. Today, I'll be talking to you about our wonderful orchid collection and what we've been doing with it over the past couple years. So to start off, this whole project with the orchids began in 2022 when Seth Hamby started doing inventory of the orchid collection just to see what we had, focusing on all those orchids that had labels on them. And this is something that I continued in 2023. And then we also took pictures of any blooming orchids that were unidentified and tried to assign IDs based on the flower morphology. And then that got us up to about 800 orchids. And that's something that we finished this spring in 2024. And that was just the basic step for curating the orchid collection. After that point, we started to look at some of the plants we had and some of their health symptoms. We noticed that a lot of the orchids had these necrotic patches on the leaves. And so I kind of did a little bit of a dive into what different orchid diseases are out there, and some resources, some excellent resources are out there. So I really appreciated the photo galleries from the St. Augustine Orchid Society. And then also the American Orchid Society hosts some talks by Dr. Ron McCatton. And these are talks that are recorded, so you can go afterwards and watch them. And it's really great because people will will send pictures of their diseased orchids, and he'll talk about likely pathogens and treatments as well. So this is the St. Augustine Orchid Society photo gallery specifically for viruses. And these are the symptoms that visually, at least to me, I was like, okay, I think we have this. So after that point, I wanted more confirmation than that. So we decided to we also decided to move on to testing. But before any sort of procedure for testing, we wanted to make sure that anything we did, we weren't going to spread any more of this potential virus. So we looked at various protocols that were out there for testing, for cleaning orchid equipment. There was this protocol from Anna Sadara, originally from Dr. Kate Klausing, which involves soaking pruners to disinfect them. And according to them, this was more effective than flaming pruning shears or using some other commercial chemicals that are out there like Physan. So in this protocol, the first thing you want to do is rinse off your pruners of any vegetative remains, any leaf beds that might be on the pruners with water. You'll soak in lye for at least 5 minutes, rinse again, and then soak in a solution called Virkon S for another 5 minutes. And the idea is that if there are viruses in the plant that you just pruned, the NOH will break apart the viral capsid protein, and then the Virkon S is something that destroys nucleic acids. So you should have pruners that are ready for your next exploit after that. And this is something that you really have to step back and think about before you do any sort of curation of the collection, because the viruses that I'm talking about today are systemic. They're present in all plant tissues. It's something that you can spread whether you're using pruning shears to divide back pseudobulbs and make new plants. Maybe you're gonna cut your Phalaenopsis orchid to stimulate a secondary bloom spike. Maybe you're going to trim roots or offsets. Any of those activities could potentially spread systemic viruses. So after the initial visual confirmations of viruses, we actually ordered some commercially available tests from a company called Agdia. They offer a product which will test for Cymbidium mosaic virus and Odontoglossum ring spot virus. And you can simply place the strip into a solution with the macerated leaf. And the first 5 tests that I completed were discretionary. I tried to choose some which were obviously virused and some which were obviously not. And I found that 4 out of the 5 did test positive for one or the other. For the next 20 tests, we assigned them randomly, and we found that about 60% were positive for one or the other. So at that point, I was confirmed it's okay that we can go ahead and buy more tests because we have a lot of orchids. So I want to talk a little bit about the actual viruses themselves. I mentioned a little bit about the symptoms, which are similar for each virus, but they can also differ according to cultural conditions and, of course, what type of orchid it is. So Cymbidium mosaic virus and Odontoglossum ring spot virus have been known in horticulture since the 50s. So it's something that collections have been dealing with for a long time. They have similar genome sizes, and they are both RNA viruses. So once they spread, they can immediately start creating new viruses once they're in new plant tissue. And then, as I mentioned, these are both systemic viruses that primarily spread through pruners. And here is a little bit of a visual. On the left, I have ORSV, which has kind of more rod-shaped um, uh, viral capsules, and then CYMV is more flexuous, they say. And then besides orchids, there are some other plants that these viruses can infect. Um, However, most of these are test organisms that have been used to study the viruses, especially things like tobacco. And then I wanted to talk a little bit about the genomes of these viruses. So both viruses have parts of the genome that help them replicate themselves. So they have some polymerases. Once they get inside a plant cell, they still need to be able to move between plant cells and also throughout the whole plant organism. So there's proteins that help them do that. And then there's a coat protein, which protects the viral genome and then also can assist in cell-to-cell movement. And then there's this technology called viral-induced gene silencing, which can actually is a technology used by people to create strains of orchid that are resistant to these viruses. And these are going to depend on natural plant defense pathways. So plants have mechanisms to silence viral DNA. And if you create transgenic plants which overexpress certain coat proteins or double-stranded nucleotides, then you can kind of ramp up the plant defense mechanisms and create resistant plants. Of course, this is only done in cultivars of orchids that are really important for floriculture and for potted plants. It's never going to be the solution for more collections-based endeavors. And then, of course, the plants have tools, but so do the viruses. They produce proteins which can destroy some elements of the plant defense mechanisms. And then some of these elements can also assist other viruses. In this study, they inoculated Phalaenopsis orchids with viruses on the leaf tip. So they worked with both of the viruses that are that we tested for in our collection, RSV and CYMV. So when they infected the viruses separately, they observed the virus in the plant tissue at a certain time point, but then when they infected them together, you actually see right here that one of the viruses spread further at the same time point when it was in the presence of another virus. So the ORSV actually helps the CYMV spread within the plant tissue faster. And then so ORSV is the one that had this protein that helps movement of other viruses and also combats some of the plant defenses. And so this is the reason why transgenic lines have only been effective for Cymbidium mosaic virus and not Odontoglossum ring spot virus. So I mentioned in the beginning that it was really great to have visual resources to kind of get started, to confirm the problems we had. So that was something that was really important to me throughout this whole project, was to document both the symptoms and then also the results of the tests. So for each plant that was tested, I have in the Living Collections database associated with that accession a result of the test. And then I'm going to go through just to show you that these viruses can affect pretty much any sort of orchid that you may have. So here we have some Cattleyas. These have necrotic patches right here, but some orchids may not express any symptoms whatsoever. So that's why the protocols you set in place are so important. So moving on, we have cotyledons, dendrobiums, vandas, oncidiums, paphiopedilums, stanhopias, catasetums. And as you can see, the symptoms all present slightly differently. So it's hard to go by that alone, which is why the testing was so useful. This is a summary of which orchids we had at the beginning of the inventory. So the American Orchid Society classifies orchids into different groups called alliances, which is mostly based on which orchids are related to each other. You can see that in the beginning, we have mostly Cattleya alliance orchids. And then this is a diagram of the eventual fate of those orchids. And I'm going to, from here on, just focus on the ones on the subset of those that we tested. Here's a bit of a close-up of those tests. And also, I just wanted to mention that the subset of tested ones that actually had good results was a little bit smaller than the number of tests we bought, because occasionally there would be issues like the control bar not showing up. But for the most part, that wasn't too much of a problem. For the final results for those subset of 788, we had 51% positive for Cymbidium mosaic virus, and then 26% positive for Odontoglossum ring spot virus. And then if you consider both viruses, we had about 60% that were positive for one or the other or both. Here's the breakdown by alliance just for that tested subset. And then here's a more fine scale diagram of the results. So in the along the x-axis, we have the different alliances of orchids, and then the counts for each testing result. So the red is all negative, so they tested positive for both viruses. The green tested positive for Cymbidium mosaic, but not ORSV. Blue is the reverse, and then purple were positive for both viruses. And we expected that we might see a little bit different patterns between different types of orchids, which you do see, but again, it's not too clear. We still see all four results for pretty much every alliance. So this is our final collection breakdown. We have about 300 orchids that are negative for both viruses currently in our collection, and then we're still pretty Cattleya dominant. So as far as takeaways, protocols to combat viruses are really important. Primarily, you want to sterilize those pruners, but they're viruses that can be unpredictable. It's a good idea to try to combat pests that may serve as vectors for viruses, or weeds, which may serve as alternative reservoirs for viruses. And then also these protocols are going to help not just against the 2 viruses we tested for, but any other virus that may be present in the collection, unidentified in our collection, or maybe unidentified period. And then this keeping a hold of our collection like this will make it easier to share material with other institutions, material that we're confident in. And then going forward, we're going to implement all these procedures, and then also limit the scope to focus more on New World orchids. All right. I'd like to thank Austin Brennick and Megan and Julian, interns from the summer who helped with all the testing, as well as conservation greenhouse staff and volunteers, and then my supervisors for ultimately approving the project. Thank you.
We have time for one quick question, if anyone has a quick one.
I might have missed it, but what did you do with all the infected ones? Do we still have them or are they
No,
destroyed?
we did call them. Yeah.
Thank you, Ben. Next up is Dr. Alejandra Vasco.
Thank you, Jesse. Thanks, Erin, for organizing this symposium, and thank you all for coming. It's just very exciting to be hearing about all the different research topics that we are sharing with other people outside of bread. So today I will be talking about Elaphoglossum phylogenomics, perspectives and challenges in the study of one of the most diverse group of ferns. This is a talk with my colleague Weston Testo from the University of Vermont. And this is a talk I was invited to give at the Symposium, Fernal Lycophyte Evolution, a phylogenomic perspective that happened at the International Botanical Conference this summer in Madrid. Here are some of us. It got very well represented. A lot of us came. And here you can see some of our interns or students that also have come to work with us. So it was very exciting. And it was also very exciting for me because for the first time I had all my grad students that are co-advised throughout the world together. Some of them haven't met each other personally. So it was very special. So coming back to Elaphoglossum, so this is a lot of people are working on this project, and you can see here all of them and their institutions. And so I want to give a quick look at Elaphoglossum. What have we learned in the past 50 years, lessons from new data, and the next steps? So how can we recognize Elaphoglossum? Like I say, Elaphoglossum is one of the most diverse genera of ferns. It has simple entire leaves, so sometimes it doesn't look like a fern. So that's what is called Elaphoglossum. Elapho is deer and glossum is tongue. So it's the deer tongue fern. It has dimorphic leaves, meaning that the leaves that are fertile are different, shorter, longer than the sterile one. It has acrostichoid sori, meaning that the sori cover the whole undersurface of the leaves. They have philopodia. There are these dark structures at the base of the petiole, free veins, and 60 to 80% are epiphytes. So Elaphoglossum is one, as I said, it has more than 600 species, so one of the most diverse fern genera. It grows principally in tropical America, as you can see here, but it's all like in the tropics, with another center of diversity in Madagascar and Southeast Asia. It's principally montane. So many species. So maybe you were thinking that I was going to talk about the ferns of Colombia. That was another talk I gave at BC, but I decided, like, I've been talking here too much about this project, the ferns of Colombia, but there's so many species. And this is just one example that, so this is the checklist of plants and lichens of Colombia. And as you can see, Elaphoglossum is one of the genera. It doesn't matter, like all of these are flowering plants, but it's the genus that have more species of in Colombia of ferns, but it also ranks among the genera with more species. So here I'm with Lucia, my grad student that is here in Colombia, where we're just looking at the collections there. And this is what is saying, yeah, this is the bane of my existence. So we are collecting this very diverse genus, but the species is very difficult to identify the species because there are not many characters that can be helpful to distinguish them. So in 1980s, there was a classification based on morphology published by John Mickle and Lucia Tortua. And this is the most recent classification that we have. It divides the genus in 9 sections, 20 subsections, and it's very important because it gives a key to sections and subsections. So if you have a new species that you don't know where it goes, you can use the key. So it provided coherent set of characters, and this has been the basis for all future works. This was only morphology, right, in the 80s. We entered the molecular data for Elaphoglossum in 2004 with a couple of phylogenies published mainly by the people here and collaborators. And the phylogenies were really exciting at that time. That was 20 years ago. I was starting my PhD because they kind of like the main groups that we have defined on the basis of morphology or that were defined on the basis of morphology were being recovered. So we started cleaning up clade by clade. These are the monographs that some of us has produced. And there have been also regional treatments. I brought this for MIADS. This is the treatment that was done on the Madagascar Elaphoglossum with 23 new taxas, 22 of them were endemic. There has been also a lot of regional treatments. So a lot of work has been done, but almost 90% of the genus is not in monographs, so it needs systematics reviews. Lots of new discoveries. We're discovering a lot of new species, and we have many complex species complexes that we haven't, we need to, to understand, but we also are having very limited resolution in chloroplast phylogenies. So we have a long way to go. So what we're doing now with the Elaphoglossum is, as many people actually in IBC show, we are getting phylogenomic data sets. And in different community, we have a probe set that is called GoFlag that was designed through a big NSF project of all the flagellate plants. So this is a probe data sets that give us around 400 loci for normally exons and flanking regions. This is very similar to 353 angiosperm data set, but it's mostly for ferns and all bryophytes and all the other flagellate plants. So this is what we are having. We have right now for Elaphoglossum 233 accessions. We are doing this, we are running them with HiPiper from Matt Johnson, the software that he developed for 353. It's working fine for this data set. And we are also doing something that is very cool. Hybrid phylogenetics is very interesting, but it's very challenging, but we are using hybrid detection with high phaser through variant phasing. I lost track of my time here. So after all of this, this is expensive, and this is a lot of time analyzing. We are getting a phylogeny that it's very consistent with the chloroplast phylogeny, in which we have here the sections mainly defined by morphology, but we have much better resolution. So that's just very exciting. With this chlorop data set, we're getting lots of loci from sample with very little DNA. So that's been really great too. We can use herbarium specimens. And so as you can see here, we have the total DNA and the number of row reads. So we can get a lot of reads with little DNA, which is great. So we are finding different things. We have found that some species have different positions in the chloroplast and in the nuclear phylogeny. And this was, for example, one species, that Elaphoglossum ducei, that when we saw the chloroplast phylogeny, we were surprised because the spores, it was in this group. In the chloroplast phylogeny, it was recovering this group. And the spores really didn't look like them. So in the nuclear phylogeny, we are finding it in a different place, which is probably suggesting hybrid origin. So there might be two species belonging there. But so that's exciting. And we are looking now, this is Ana Gabriela's work, and she just published the taxonomic revision of these species. So with her, we are having the possibility of sending more samples of a single species and seeing if the resolution we are getting with these 231 accessions is going to be the same. And as you can see here, we still have some problems. So not problems, but species that didn't came in this phylogeny, which is with this GoFlag data set, didn't came in the position that we thought they were going to come. And we are, with Anna, are looking into this. Are these really misdeterminations that we didn't see? These groups is this group again is very complex. The characters that differentiate the species might be very subtle. So she's going back to the herbarium and looking at these specimens again, but it might be also that it's showing some hybridization. So we're looking into that. And here is the biggest analysis we did with HypePhaser, looking at variant phasing and assignment. And so here, I know it's, like I say, I can spend an hour talking about this analysis, but what I want you to see is that this is the phylogeny that we produce, the phylogenetic hypothesis. And here you see two columns with colors. So these two colors are the locus heterozygosity and the allele divergence. So when you see colors and dark colors here in both lines, that's probably showing that we have hybrid or polyploids. So here you can see that if you graph those two, this tail here where you have a high percentage of allele divergence and a high percentage of locus heterozygosity are the putative hybrid taxa. So the first time I saw this result, I almost ran away and said, okay, I'm not gonna do any taxonomy again. There's just no way. We have around like 20% of taxa that are probably hybrid origin. But we are looking at this data set again with the morphology. And here is like to look, this has been very exciting to get because this is how Elaphoglossum looks like. And we have them growing all over the tropics in the mountains where we mostly do field work. But how would a hybrid between these species or this plant and this plant look like? It's very difficult like to really understand them morphologically. So now we have this data set that is telling us that we have all these hybrid and polyploids. It's not a surprise. We were expecting this, although we didn't know much about these processes in tropical ferns. But now we need to just go back to the collections and really look how we can figure this problem out and kind of keep studying the diversity of these big genera. And so we are also putting together this occurrence data set to summarize the Elaphoglossum diversity. So to kind of like have a bigger database of the collections that we could be using to kind of increase the sampling for phylogenetics. So we are looking at species richness, collection biases, quality of determination. And we are using mostly GBIF and Torito portal, which is a Symbiota portal. And we got 88,000 records. We did name resolution with these 2 R packages, Torito, Cat, and Taxland. And so here you can see one of the results that we got. This is the global Elaphoglossum diversity. Guess where Colombia is? Look at that yellow. There might be some bias there in terms of collections and determinations, but you can see that this kind of follows the pattern that we have found before, that 75% of the diversity occurs in the American tropics. And we have there Ecuador, Costa Rica, and Colombia, and Peru as the really diverse. But look at the states of the collection. So we do have a lot of indents. They're here. I just don't know if you can see this thing. Yeah, the indents, the percentage of indents is really high in this tropical, in the most diverse countries. So a lot of work to do taxonomically and with the collections. And I wanted to highlight Venezuela and Panama, where we really have very low collections, number of collections per species. So there are 2 tropical countries, and there are many reasons why we think that might have happened, but we need to do collections. If you ask me, where should we go to collect Elaphoglossum, I think it should be Venezuela and Panama. So the next steps is that we are dealing with the species complexes, and we probably have reticulate evolution. That means hybridization and polyploidy happening, but we want to do complete data sets for this probes so that we can tackle that. And this is something that Lucia is doing in her PhD at TCU. We are doing the taxonomic revision. We need to do the taxonomic revision of the big sections, and here we need some volunteers. I'm doing the Lepidoglossa, but section Elaphoglossum needs someone else. And we're working on, this is the next thing, is that working on macroevolution, maybe Lucy, right? Niche evolution and range dynamics and biogeography. With that, I just want to say thank you all. I know this was very fast and this was for an audience on like fern people, but I wanted to bring it here and I will be happy, to discuss a little bit more about Elaphoglossum and the methods. And here is this video. So Lucia gave a poster also on IBC, and here you can see her video talking about the poster, and she's going to put the poster available in case you want to learn a little bit about what she's doing with this data set with her group. Thank you. Do I have time for questions or do? Oh yeah, look.
Yes, we have time for
15 minutes.
questions.
I really thought so.
Very interesting talk, very impressive. I know you've done some SAM work. I'm interested in knowing if scanning electron microscopy also gives you data to identify species, and how does that correlate with the molecular data? You didn't say too much about ESSA yet.
No, I didn't say. So Elaphoglossum, like I say, is a genus that has a lot of species and very few subtle, very few and subtle characters. Most of the taxonomy is based on millimeter long scales, so that and spores. So we use a spore micro-ornamentation, which is the ornamentation you can see when you do scanning electron microscopy, to also help us with the taxonomy. So we use it all the time. And actually the classification of the 80s that was proposed by Lucia Teortu and John Meikle, one of the characters they used to differentiate those groups were the spores. We are actually now having one of our interns, also funded by the NSF, Grace Pascarella, that is working on one of these clades, and she's doing SEM to try to better understand the evolution of the spore ornamentation. But yeah, we use it all the time. It's very useful. And we have found that is the morphology of the spores is very consistent within the species. So it helps us, because from some genera of lycophyte is not. So, but we know that in Elaphoglossum, it's very helpful. Thank you.
If you're good with microscopy, you can relate what you see with the microscope with the SEM, and you can actually see those characters with the microscope. And so it becomes more valuable if you can make those correlations.
Correct. Yeah, I think this is something that also actually Grace is doing, not only looking at the spores, but also putting them, the characters, coding them in a matrix so that we can look at their evolution. Yeah, thanks. There was another question, Brooke.
Thank you, Ale. Great presentation. Can you talk a little bit about how hybridization in ferns works? So they do have that separate gametophyte generation. So is it something where it can go, do they still have any concept of selfing or outcrossing, or is everything in a certain genus, anything in it, can they completely cross? Is everything viable within a genus or across genera? Because it's really different than with flowering plants.
Yeah, that's a fascinating topic. We don't know much in Elaphoglossum, so I cannot tell you if everything hybridizes or. But in ferns, we know that plants do hybridize, and they can double their genome and become fertile, and they can outcross. And so they can do everything, and they are happy, and they actually, when they hybridize and double their genomes, they get stronger. So plants are amazing. And we know that in ferns, the percentage of hybridization, at least, or species that whose origin is hybrids, is way higher than angiosperms. I didn't bring that slide, but I think it's like 20% of the ferns are of hybrid origin when you look at a world scale. And then I think 10% or 12% of angiosperms are. So these are things that are happening all the time and that really have shaped the evolution of plants on Earth. It's just that for phylogenetics, we kind of like wanted to forget it because that makes things very difficult. But in ferns, as you were saying, so ferns have two different plants. They have two different phases that they are independent. So the sexual phase, that is the one that produces the gametes, and the asexual phase, that is the one that produces the spores, they live independently. And so the barriers for fertilization, or like if you have a sexual phase of one species living with the other ones, that's called gametophyte. If you have two gametophytes, they only need water for one to go to the other. So those barriers are a little bit lower, and that's why we think it might be that hybrids have a higher percentage. But no, when we look at genera that are very well studied in the temperate regions, if you think about male ferns like Diopteris, we know that not all of them hybridize with all of them. And so that might have to be with many different regions. One is like if you are not distributed in the same place, you cannot hybridize with the other. But even when things are living within a certain region, some species do not hybridize. And it can be that they are like the sperm mobility, as Eddie was talking in one of the talks here, maybe you have a sperm that doesn't go too far away, so you cannot hybridize with others. There are asymmetric hybridizations, so we know that some species are always the one that provides the egg, and some species are always the one that provides the sperm. So there are many ecological and physiological issues that might be happening in the different species. In Elaphoglossum, we don't know anything. There have been some hybrids described because we have some species that have divided leaves. So they hybridize. For all the species that have divided leaves in Elaphoglossum, there are 6 species, we know hybrids. So I bet you all these species that only have simple entire leaves are all getting crazy and hybridizing, but we don't know much. And this was once actually one of the main messages of this symposium at IBC, is that a lot of people that are working with this big genera of ferns, mostly they are in the tropics, but some of them are in the temperate zones, is that we are seeing a lot of hybridization and polyploidization in this, I mean, at least the signatures of those in these data sets, and we have to start looking at them. So, long answer.
Thank you, Ale.
Thank
That's
you.
all the time we have for today. Let's give another round of applause to all 3 of our speakers. Our next lunchtime lecture is November 12th. You can look forward to a similar format of a variety of talks from our research staff. Thank you all for coming.